Week 3, Principles of Clinical Cytogenetics and Genome Analysis, Part 2. If we look at atypical chromosome structure, we see that a chromosome can be structurally atypical in several ways. There can be too much or too little genetic material. Some of the DNA may be inverted. Some of the DNA may have been moved and inserted to a different type of chromosome. Atypical chromosomes are balanced if they have the normal amount of genetic material and unbalanced if they have extra or missing DNA sequences. If we look at the hypothetical normal gene sequence, it appears, it can be shown in A, normal sequence of genes. If we then look at B, we see that there is a deleted sequence of genes. And in C, there is a duplication. And in D, there has been an inversion. A deletion is where we are missing a genetic segment from a chromosome. They're often not inherited, rather they arise de novo. Larger deletions increase the likelihood that there will be an associated phenotype. Crete-Shah syndrome, cat-cry syndrome, which involves a deletion in the 5th chromosome in the short arm, is something that we will be looking at in the next unit. Duplications, the presence of extra genetic segment on a chromosome, often not inherited, rather they arise de novo. The effect on the phenotype is generally dependent on their size. Larger duplications tend to have an effect while smaller ones do not. This next slide illustrates duplications in chromosome number 15. A study of duplications of parts of chromosome 15 revealed that small duplications do not affect the phenotype, but larger ones may. The letters indicate specific DNA sequences which serve as markers to compare chromosome regions. Note that the duplication is also inverted. Translocations. This is what takes place when two non-homologous chromosomes exchange segments. There are different types of translocations. We have a Robertsonian translocation and reciprocal translocation. In the case of the Robertsonian translocations, two non-homologous acrocentric chromosomes break at the centromere and their long arms fuse. Short arms are often lost in this process. This affects 1 in 1,000 people. Translocation carriers have 45 chromosomes. They produce unbalanced gametes. In the case of the translocation that can take place in Down syndrome, about 4% of individuals born with Down syndrome results from a Robertsonian translocation between chromosomes 21 and 14. This tends to recur in families who also have a larger risk of spontaneous abortions. One of the parents is a translocation carrier, and they may have no symptoms. And there's a distribution of the unusual chromosome, which leads to various imbalances. I'd like you to take a look at this slide showing a Robertsonian translocation. In this case, Mr. P has only 45 chromosomes because the long arm of one chromosome 14 has joined the long arm of one chromosome 21. He has no symptoms. Mr. P makes 6 types of sperm. A sperm with one normal chromosome 14 and one normal 21 yields a normal child when fertilized. B, a sperm carrying the translocated chromosome produces a child who is a translocation carrier like Mr. P. C, if a sperm contains Mr. P's normal 21 and his translocated chromosome, the child receives too much chromosome 21 material and has Down syndrome. D, a sperm containing the translocated chromosome and a normal 14 leads to excess chromosome 14 material, which is lethal in the embryo or fetus. If a sperm does not have the chromosome 21 as an E or 14 as an F, a monosomy results, which is lethal prenatally. Reciprocal translocations are where two non-homologous chromosomes exchange parts. 1 in 500 people are carriers, usually healthy because they have the normal amount of genetic material. The translocation breakpoint interrupts a gene. There may be an associated. In this slide of a reciprocal translocation, the two non-homologous chromosomes exchange parts. In A, genes C, D, and E on the blue chromosome exchange positions with genes M and N on the red chromosome. In B, a reciprocal translocation has been highlighted using fish. The pink chromosome with the dab of blue and the blue chromosome with a small section of pink are the translocated chromosomes. Inversions are where chromosome segments have been flipped in orientation. 5 to 10% cause health problems, probably due to disruption of genes at the breakpoints. There is a pericentric inversion. This is where the inverted region does not include the centromere. Pericentric inversion is where the inverted region includes the centromere, and it may impact meiotic segregation. Here we have a slide showing pericentric inversion. A pericentric inversion is where one chromosome leads to one normal chromatid, one inverted chromatid, one with two centromeres, dicentric, and one with no centromere, an acentric fragment, if crossover occurs with the normal homologue. The letters A through G denote the genes. Here we have pericentric inversion. A pericentric inversion in one chromosome leads to two chromatids with duplications and deletions, one normal chromatid and one inverted chromatid if a crossover occurs with the normal homologue. Isochromosomes. Isochromosomes is the result of another meiotic error that leads to unbalanced genetic material. It is a chromosome that has identical arms. An isochromosome forms when during division the centromeres part in the wrong plane. Isochromosomes are known for chromosomes 12 and 21 and for the long arms of the X and the Y. Some women with Turner syndrome do not have the more common XO abnormality, but have an isochromosome with the long arm of the X chromosome duplicated, but the short arm absent. If we look over here, we can see that the isochromosomes have identical arms and how they form when the chromatids divide along the wrong plane. In this depiction, horizontally rather than vertically. Chromosomes shaped like rings form in one of 25,000 conceptions. Ring chromosomes may arise when telomeres are lost, leaving sticky ends that adhere. They can be caused by exposure to radiation, and any chromosome can form a ring. Most ring chromosomes consist of DNA repeats and do not affect health. And here you can see in this diagram how a ring can form. If we look at some of the ring chromosome syndromes, these arise de novo. All of them involve a microcephaly, a small head, learning disabilities or intellectual disabilities, slow growth, and unusual facial features. And the chromosome syndromes involve chromosome 9, 13, 14, 20, and 22. This slide summarizes the causes of chromosome aberrations. In the case of uniparental disomy, UPD, this is where you have the inheritance of two chromosomes or chromosome parts from the same parent. It requires the simultaneous occurrence If you look at this slide, you will see the woman with cystic fibrosis inherited two copies of her mother's chromosome 7 and neither of her father's. Unfortunately, it was the chromosome with the disease-causing allele that she inherited in a double dose. Amniocentesis and chorionic villus sampling provide a prenatal diagnosis because they directly detect chromosome abnormalities known to cause specific clinical conditions. Here we see amniocentesis. It detects about 1,000 of the more than 5,000 known chromosomal and biochemical problems. Ultrasound is used to follow the needle's movement. In chorionic villus sampling, it's performed during the 10th to 12th week of pregnancy. It provides earlier results than amniocentesis. It does not detect metabolic problems. It has a greater risk of spontaneous abortion. If we look at the indirect detection of chromosome abnormalities, what they do is measure or detect a characteristic that is associated with having a chromosomal abnormality, and they're used as screening tests to identify individuals at elevated risk. They do not provide a diagnosis. Maternal serum markers are being replaced by testing fetal DNA in the mother's bloodstream. This is non-invasive prenatal diagnosis or testing. This can be done about 10 weeks into the pregnancy or later. This is the end of week 3. Please complete the karyotyping exercises and complete the 4 questions. Please submit question 4 to me for grading. There will be a test at the end of week 4, and it will contain all of the material assigned in the weeks 1 through 4. Week 5 will be a holiday week for Thanksgiving, and you will not be given any new material.